we as a field need the database or the combination of databases, and we need to have the discussion as a field to determine what it is that we need moving forward. And so we have the right people in the room to start that discussion right now. So I'm going to pull some of the questions. We're going to shift over to our panel discussion, of course, at this point. And I'm going to pull some of the questions that have already been asked in the Q&A, but by all means, please feel free to type new questions. And panelists, you can either type questions in the chat so that I see them, or you can ask out loud, but we're just going to go right for it. And so for the panel overall, how do the mined databases deal with contaminants? And so, for example, the example that this person asking the question has given is that, we deposit our raw reads into public databases, but of course, when we're, analyzing our data, we're curating those in some way. We're assembling, maybe we have, a negative control, and we're actually culling some of those reads in our analyses. But then if you go back and you mine, say, SRA, how do these various databases handle those kinds of contaminants, or do they?
I can go if you want. I mean, so the way that we try and position everything is we try and give as much information as possible. So if we're using CheckV, when we'll be reporting those results of the CheckV, or if we're using bacterial bags, we will use CheckM and report the quality scores. So one thing is to have the information. We also tend to be quite conservative. So we, in terms of the human gut phage database, we could have claimed much bigger numbers if we didn't use such a high number, in terms of quite a stringent threshold of wanting contigs longer than 10 kb. So we always tend, my world always tends to err on that side of caution in terms of when we're reporting. It's also the same when we're clustering genomes. We tend to cluster we tend to choose something where we won't over-report numbers. I think the other thing that Magnify does is we try and have everything linked back so that you can try and trace that contamination. We know we're going to make mistakes. We know that there are going to be contaminants that leak in. And ultimately, if we've got that full provenance back from, contigs, assemblies, raw reads, we've got the opportunity that if there's a new fancy assembler that overcomes all of the issues with potential chimeric sequences, specifically with viruses in mind, etc., I think that's the best we can do, because we want to try and give that wealth of information, be conservative, but also allow people to go back. And I think that's the way that we try and handle it as best as we can. I would say that we're always limited. So the number of people who are on production in my team, Magnify is run by sort of 6 people. That might sound a lot, but it's not very many people when you look at the volume of data. So we can't do anything manual. So we have to rely on tools and best approximations. And when we're talking about the scale that we're looking at, we have to do things computationally. And there are a couple. One other point I need to make is, when you're running a production service, we're always slightly behind the bleeding edge of research science. So it takes a while to get tools in place and really, well, certainly Magnify is, but it takes a while to work out that a tool's really good, it's accepted by the community, and we should introduce it into a production pipeline and then make it available for everyone. And there's a lag, and sometimes people go, oh, Magnify should be doing this. It's like, it just takes a bit of time.
Excellent, thanks. That makes a lot of sense. And I should say also, to clarify, that this was sort of a multi-part question, and it would include contaminants that are known, say, in reagents and things like that, and even Phi-X, for example. And so on top of curated data sets, which of course the user is going to try to curate themselves, and that might be missed in the SRA, you might have these contaminants that get missed. So Simon looked like he wanted to spring on that. Yeah.
Yeah, I think there are multiple tiers here. One aspect is it's different if you're running a database that you create on your side, and if you're running a database which is basically user contributed. The former being what is done right now for UVigs. If you look at IMDBR, if you look at Magnify, the viral contigs kind of database part, it's really, you know, us or API kind of going in and deciding that this is your list of viral contigs. And as far as I know, and Rob can, you know, contradict me if I'm wrong, but I don't think people can come in and say, well, these are virus contigs that I want to add to Magnify virus contigs, and, you know, you just accept them blindly. We're not there yet, basically.
So I mean, the aspiration is that you could add that into the layers. So that's sort of a bit like the mags. People can submit their mags in. We'll submit our mags in. We'll take the consensus of of that set, but that's not there yet, as you say.
Yeah. So in one part, in one side, it actually takes care of some of the issues like you were mentioning, Joanne, because basically, at least there is a standardized pipeline that is applied, and we are trying to avoid the obvious contaminants. Phi-X are basically flagged everywhere, stuff like this, because we still have it. I don't know why, I don't know how. It still keeps popping up.
Well, why is easy to answer.
yeah, no, I mean, like the actual contaminant Phi-X, not the real Phi-X.
Yeah.
But anyway, I think then for contaminants that we know exist, but we don't know exactly which virus they are, like, you know, new contaminants get detected all the way in reagents, for instance. I think this one will be more tricky, and we will have to eventually, you know, get a better handle on this again, probably through a common database. I don't think that this exists yet. But at least for the UVic database that exists right now, you have this layer of like, there is some gatekeeping, for better or worse, there is some gatekeeping being done, because it's not yet a fully user-contributed database like SRA. SRA is much more for like, people also can submit raw reads or filtered reads, and it's really hard to kind of know which is which. So.
Yes, and actually it's not only a contamination problem, it's also a problem of wrong labels to what already is in existing databases. And I think this factor can be even larger sometimes, at least at the moment, very often. If you try to download something, it is just not correct. Like complete genomes are not really complete, 5' and 3' UTR are missing, the same or similar for phages, but also a lot of mislabeling. And I think it's important for a user also to be aware of that whatever you download, that there is quite a huge fraction wrong annotated. It is, I think, important. And I think also this is something, it's our job to clean up in the future, for sure. Yeah.
Plasmids are not plasmids, they are phages, stuff like this. Just wanted to throw this out there on record, because we have this so much. So just so everyone knows, don't take plasmids from like EBI or NCBI and assume all of these sequences are proper plasmids. A bunch of them will be actual phages. So don't use these sequences to test if the plasmid detector works well, because.
Exactly. But that happens so often. So this is, yeah, it makes us smile also.
That's right. Right. So there's some kind of follow-up questions that are a little bit related to that. So I'm going to take those before we move on. So can we also subtract UVigs and MAGs? So let's say we want to know something is a kit, we know something's a kit contaminant and it's found in soil virums. Can we flag that? And I guess the question is probably, to what extent does, you know, the user need to flag that on their own after they say download or interact with the database? And to what extent is that taken care of, or can that be taken care of on the database side? And Evelyn, please feel free to speak up if I'm butchering your questions. Thanks.
So I maybe I can answer to that, and it's something that we've toyed with a lot. It's very hard to do at scale in a automated or semi-automated way. So in the ideal world, what we would do is when someone submits, I'm coming from the metagenomic point of view, so just excuse that caveat, but if someone submits their study, if it was clearly labeled what was a negative control in the metadata and it was deeply sequenced, then we could get the kit contigs out. And I've done that with so the skin study that was done in collaboration with Julie Segre's lab. She had amazing controls, and we could really identify what was contamination and what wasn't. And that was really good, except on one of the controls, we had a Corynebacterium, which has probably come from someone in the lab handling the Corynebacterium, and it's got into handling from the sample or the test tube or whatever. I'm a bioinformatician. It comes off the sequencer, but essentially it's probably a lab technician contamination. If we subtracted all the Corynebacteria, we would be slated because we're missing Corynebacteria, which is the well-known skin. So it's very difficult to do without really having a really deep knowledge of the data set, and it's quite difficult. So we both lack the controls at the first place, and then to automatically do it is a bit difficult.
All right, great, thank you. Oh, Simon, go ahead.
It will feel like a cop-out, but I don't think this is a database side of the question, just because the best way to detect contaminants, and I think that's what Rob just explained as well, you need to understand what you put in your sample. You need to understand what group of samples were processed together. You need to understand what contaminants you expect and what's not, like what actual biological signal do you expect to really identify. And that's when the database gets the data, it's too late. I know there has been some efforts at one point to have all this information stored into the database, the batch run and the sequence and, where it was sequenced and everything. It still makes it way too hard, I think, on the database to really find contaminations. And so I think it's on the user side on this question, yeah.
Yeah, I agree, despite being a user and thus now being responsible for doing that, but I agree. All right. So we'll move on. There was a question about databases or resources that would be good for just a standalone nucleotide blast of Virum data sets specifically. So if a user wanted to blast against a Virum data set, either on the web or wanted to download a data set that they could run their own blast against, what would be a good data set for doing that for Virums?
Well, maybe I can talk about what we are doing like daily, but we are also bioinformaticians and it's going probably into a different direction. But what we do is actually we just download everything related to viruses from every source which we can find at the moment. And then, we are not working on phages, so maybe I need to be careful what I'm saying. And then we try to work on that. And actually BLAST is too slow in order to really make a comparison. And usually we use different tools than BLAST for doing that. Most of them can be K-mer based, can be also something else. But I think most of the classification tools also work K-mer based, like Kraken and other tools like that. So I guess this is a kind of current thing to do. I don't know, maybe Rob, you want to complement that?
It's a bit a similar question, really. It depends what you want to do. What's the objective of running the blast would be I would bet, because that should dictate the database you should use. What are you trying to achieve? And so that's what I would say is I'd throw the question back and say, what are you trying to do?
I was actually thinking that the question was classification, because that's why somebody asked about the blast. So I was thinking it was classification, but maybe it's not.
But if it's classification, I think I wouldn't use BLAST either. But I think there are some databases coming out like the ViFogs or the VOGs databases that can give you sort of diagnostic if it's to do with classification as a first pass. And then if you really wanted to do something even more fine-grained, then I would probably look at, your best bet is something like NCBI with the full sequences or the high quality out of IMG VR. Sorry, I'm probably butchering.
Yeah, I think I would agree with that. And I maybe also, I'm not a user who does exactly that, but I did actually, in preparation for the database introduction yesterday, I just tried taking a random viral sequence and running a blast search on NCBI's online tool. I honestly can't even remember exactly where on NCBI I found this thing, but it's somewhere on the NCBI sort of viral genome portal. You can actually run a blast against their curated various parts of their curated data set. So not just like all of NR, but actually virus specific. And for my unknown sequence from soil viruses, I got 0 hits. So depending on your question, that may or may not be useful. But yeah.
Yeah, maybe I can also add that Arvind, one of our panel people, was also just pointing out that actually maybe it's sometimes worth to not only check a virus database, but actually all sequence databases, because sometimes it can happen that something has been labeled, as we said already, as a bacterial plasmid or something, but in real it was a virus, and therefore you want to maybe also check that. Yeah, it is of course also important to do. I think in general, if you think about a future direction and we want to identify our viruses, it's important we go a little bit away from always comparing to existing or known sequences from the database, and maybe instead trying to classify in a different way. There are already approaches doing that, but I think we should make our mind a little bit flexible to not only think to always compare to what we know already, but instead try to tackle it also in a more general way.
Great. All right. So this was a question that I believe has been answered a little bit in writing, but I thought it would be good to throw out to the panel anyway. So this is actually related to assigning metadata from a database to an unknown sequence. So for example, this is kind of maybe related to a V-contact analysis, but you could imagine any kind of analysis that might link an unknown virus in your data set to something known. And of course, the thresholds of that linkage, could be all over the place, but to what extent is it okay to, link your unknown virus to something that's in a public database and to assign the metadata? So that could include hosts, that could include taxonomy, that could include, any number of things. What are kind of the thresholds, and what are you looking for that match to be a solid match so that you trust the metadata?
I think this silence is telling you something. I don't know. Do you want to answer first, Simon? But I think this is a problem.
Actually, I don't know. Maybe I'm too conservative. I would not do that outside of the VOTU level. Is basically my quick answer. I'm only, and that's what we do in IMGVR, and that's somewhat based on a number of benchmarks we have done. We propagate metadata, at least in terms of taxonomy and host prediction, when genomes are near identical, this 95% ANI. And I don't, if you use 90% ANI, I personally won't be mad. It's the same thing, right? Like just near identical conceptually is what I mean here. I would not propagate host prediction, for instance, at the V-contact VC level, because we have seen a lot of V-contact VCs with genuinely kind of pretty broad host range. And basically, like taxonomy, of course, should be, okay at the V-contact VC level, again, because it's a taxonomy tool, but other metadata, in my experience of the virus world, which is mostly phages and archaeal viruses, it breaks down pretty quickly as you go up the taxonomic rank and as you go down in like similarity. So in a sense, transitivity is really limited to this identical or near identical genomes in my mind. One exception may be ecosystem, but you usually don't want to propagate this anyway. It doesn't make sense really. So I think, the metadata you want to propagate the most are taxonomy and host prediction. I would stay at the VOTU level. Maybe one thing I would do at the VC level is really broad hosts, like, maybe phylum, maybe even like gram positive and negative, because it's like the surface protein, it's super important, and it usually kind of breaks down relatively nicely at this level still. That's maybe as far as I would go.
Yeah, and I can answer a little bit as well, because this is something that we've grappled with in our lab. I mean, it's the classic balance between you just get no signal from your data set. You have almost nothing to say about these completely unknown viruses. And so you really want to grasp at straws and you know, take everything that you could possibly take into context for these samples. And so I'd say that, yes, we at the VOTU level is the way to do that. And so one way to leverage databases to do analyses at the VOTU level is, again, potentially, you know, to download the VOTU sequences and then map back your metagenomic reads. So if you don't have, you know, a VOTU that's assembled from your data set that then clusters at 95% ANI, we're talking about phages here, you can then, you know, try and do your read mapping to see if that VOTU is there. And then also this really broad scale taxonomy or any kind of broad scale metadata that you might want to add, say at the vContact level. I absolutely echo Simon, you have to be so careful. But if you are really careful and you pay very close attention to all of the VOTUs that are associated with a given cluster having the same kind of signal, for example, at a very high host, you know, predicted host, let's say every single RefSeq virus that's in your VC has exactly the same host, and you have an unknown virus that's in that VC, can you definitively assign that as the host? Like, no, but you can kind of start to make some general inferences that that class or phylum of host might be the host. If anybody wants to shoot me down on that, that's totally fine. I think it gets dangerous if you're not looking very carefully. All right, anything else on that general topic? All right, so we're gonna switch a good bit. This is for maybe people who are looking for databases. We've discussed a number of potential databases today, but are there any other sources with phage genomes, I guess, that are reasonably well curated? So kind of analogous to, say, RefSeq or GenBank, that are freely available. So the one that I thought of was PhagesDB for viral genomes, but specifically from like soil actinobacteriophages. And then I don't know if there are comparable resources from other ecosystems or human gut, or really, I'm not personally aware of a whole heck of a lot more than that. Okay. Sorry, we have work to do.
I mean, I mean, I think, you know, so we have our collection from the human gut, but I mean that there's another study from JGI that has another collection. I don't know if that's in IMG there now. So sort of, you know, the one I'm talking, Stevens. Sorry. So, but you know, I think this sort of shows you the level that we're at, and these are large scale analyses, and I don't think I'm doing Stephen a disservice, but you know, it depends what people want and the quality of curation. And you know, our data set and Stephen's data set, if you take the very high quality, they're very similar in terms of there's, you know, the numbers in totality are different because Stephen was more liberal and then did have all of the metrics. We chose a higher level initially, but are they well curated? Our data is not well curated, I'm happy to say. Yeah, it's the best that we can do at scale, because 142,000, you know, viral clusters is enough that I would not be looking through.
I think beyond the resources we kind of mentioned today on UVigs, like the PhageDB that Joanne, you mentioned, is actually an isolate database, and these are tremendously important, and we need to both like give a shout out to Graham Hatchpool and the team at PhageDB, because they do a fantastic job, and also try to emulate this, because these are like, we with our millions of virus contigs, we need this database eventually anyway. So we definitely need to work hand in hand. I think I saw Ian in the chat, the phage directory, more oriented towards like mostly kind of human health application of phages, still has and starts to compile very important resources on like which phages are available where and how. And I know this is kind of the early days, but I would say, yeah, if people are like, especially people working with human phages, but in general, people working with phages should check out the phage directory resources. It's both like community building, and it starts to have some aspect of like which data, which isolates is where, in which lab, et cetera, et cetera. And again, that's more geared for phage therapy, which I'm sure we can talk, needs specific databases anyway, and will need its own special treatment. But that's pretty much the only other efforts I could think of, which would really be a big database thing. Otherwise we have mentioned them, EBI, NCBI, we at JGI, the iVirus network, and then more kind of custom database like the Pigeon one, basically.
Great. Just a specific question here. Are there any resources for finding matched viromes and microbiomes from the same samples? That's a great question. I would also love to know if there are any. I'm not aware of any, but we should do that.
Yeah, at the moment, I think there are just people who upload their data, and then they maybe have a publication about the viruses they find and a publication about the bacteria they find, and that's it. So this is actually a problem. Maybe they link to the same data source, but there's no clear, this is a virus, and this is the data set where you also find the bacteria related to that. Yeah, at least I'm not aware of that. And I think it's also a very good point what we may need in the future. Yeah.
Great. What's the best?
Just one more quick thing here. I think we need to be careful with Virum, because I do love virums. I do love viral metagenomes. They are also not all equal. And when you look at the data, some of them really look like microbial metagenomes. I won't lie. So actually, I've been shifting towards, calling something a virum based on the data that comes out of the assembly rather than based on the pre-processing, which is maybe not great, but somewhat lets you flag out these datasets that are strongly enriched in viruses. And usually these are, for the most part, actual virums that were, meaning prepared from a sample that was, selecting for virus particles, basically. But we have to look at the data, definitely, because some of these virums, even if people have tried their best, end up looking like microbial metagenomes. And then the other thing is, yes, linking the physical sample to all the different data that relate to this physical sample is still a challenge. And I will have my little plug here for an effort that is partly led by people here at JGI called NMDC, this National Microbiome Data Collaborative, which is this very early days of how do we let people explore the data in a meaningful way where they can go from, this is a physical sample and this is everything we know about it, from like physical chemical characteristics to metagenomes, virums, metatranscriptomes, incubation with stabilizer, stuff like this. And so we are trying to come up with what would be a good framework for this. I know we have been in discussion through NMDC with all this, again, the big resources like INSDC, EBI, NCBI have all kind of stake in this. So we have been talking also with the GSC, the Genome Standards Consortium. We have been establishing standards. So like, it's a frustratingly long process by really no fault of anyone. It just takes so much time to figure out how to do this properly. But we are trying to get there, and we are not there yet.
Great. All right. So, Manja? Yeah.
I think somebody was just asking about what are the best databases for eukaryotic viruses.
Right. This part.
Maybe this part I can actually answer. This is somewhere where I'm working around. Yeah, as I mentioned already, there's this Viper where you have this human pathogen database. This is widely used for this community, which are going for eukaryotic viruses. And then you have some specific databases like for influenza, for HIV, and for some other ones. They are rather small and only specific for one virus family usually. And apart from that, it's really hard. So there's really, I mean, you have the common things like NCBI and things like that, but it's really hard to have it in a nice processed way somehow. And this is actually what we try to tackle in the future, especially if you look for viruses from fungi. So this is very difficult. Also, sometimes they don't even have a capsid and go out of the cell. So usually they even are transported with the host cell. And also for plants or for algae, it's super difficult to find literature at all, even in the form of publication. So it's really, we are really starting there. So this is a very interesting question, and unfortunately not what you wanted to hear, I guess. Sorry.
Any other comments on databases for viruses of eukaryotes? Yeah. All right, great. I mean, great question mark, right? Thank you for the answer, at least. Okay. In the Q&A, somebody says there's also Virusaurus for eukaryotic viruses. I haven't used it, but I know it exists. So there you go. All right. So one question here is, do we know the database or some source of records of bacteriophages that have had therapeutic applications in the past, for example, in phage therapy and or I believe agronomy and aquaculture? If yes, how can we recognize them? This seems like, you know, some kind of metadata that if it existed, it would be in a metadata field in the database, but I'm not sure if there's any. That sounds like a pretty specific case to me. Yeah, Rob?
And database would be the only place I'd look. I don't know of anyone anywhere that stores that level of information.
Yeah, yeah. Okay, so I think we've gotten through the vast majority of the questions that I have kind of, you know, written out. So if others have questions, by all means, please feel free to type them in the Q&A or in the chat. Oh, there's a bunch in the chat. Did I just miss something? I think we're kind of good. But I was actually just going to suggest that unless and until we have more questions, that we take the opportunity to have these panelists riff a little bit on the strengths and limitations of either their particular databases that they have already and that they're generating, or, you know, what are strengths and limitations, next steps? What do we need as a field, and what on earth can we do about it? Loaded question.
I'll start off with one. A major limitation is scale. And we can take that at every single level. So there are more metagenomes coming in than we can possibly process as you know, just at EBI. Then as we descend down, trying to really analyze those, whether you're talking gene catalogs, so you know, we have 2 billion non-redundant proteins, that's massive compared to databases like Uniprot. It's like, so that's the scale. I mean, Simon's mentioned the scale of the viruses, and how do you operate at these levels of scales to provide the dynamic range that people want? So everyone wants something different. So you might be, slicing the data that way, you might be coming down that way, and that's just sort of, you've got one, so I'll give you sort of examples off the top of my head. Someone may be particularly interested in a particular environment, in an organism that's only found in that particular environment, and wants every single example of that organism, and that's sort of slicing down. If you're trying to do, novel enzyme discovery, they don't care where it's come from. They just want all examples and just to have one gene and one metagenome. Just the scale that we're operating at, and it was sort of like the metagenome submissions that you're seeing is just dwarfing the number of isolate. And we're relatively small groups doing this who are providing these resources, and just operating at this level of scale. I don't think these are solved problems anywhere. There's lots of data scientists out there, very, very smart, but I just don't think they're solved. How we can, give this level of granularity. And I answered a question that came in the chat of why don't you provide everything and then let users filter? But then that's one user case. But then I have other users saying, I don't know what I should do. Can you tell me what I should have? And so it's, well, when everyone who establishes a resource has to make decisions, and those decisions are based on the best biological science and the finite computational resources that they have and the person power that they have. And we try and do the best that we can, but I imagine that Simon could quadruple his team. I'm sure Manja could quadruple her team, and I could quadruple my team, and we would still be having many open questions about how the heck we do these databases.
Yeah, Great. Simon?
Definitely this is a massive issue, and I guess
Agree from my side as well. Yeah.
part of the related question is at which point and how do we centralize these resources, because we will have to eventually, and I always aspire to one day have the equivalent of this INSDC, which is, as I think everyone here has experienced, this great thing which magically synchronizes between NCBI, EBI, and DDBJ, and you submit on one side, it comes out at the other side, all the identifiers look great, you have all the metadata kind of following, like it's kind of, up there as like what we should aspire to. I don't know exactly which steps we take to get there, although I would say, and again, just like organizing this kind of workshop, and thank you, Matt, and all the team to do this, it's basically one of these first steps, right? Like that's just like putting all the people in the room, keep the discussions going, and because we all eventually want to have this, all this data mirrored and better centralized, because it kind of gets a little work out of our plate to an extent. We will also probably have to have a divide and conquer approach. Since everyone wants every information on every sequence, it doesn't make sense for every single database to rerun all tools on the, so like maybe we run that thing because it's part of our pipeline, and then, using the same sequences, they are processed another way. And so we have to figure out how to kind of then reconcile and share this with the user, but I don't see another model going forward to eventually have this big resources that people can really come in and like Rob was mentioning, just slice and dice any way they want. And then you have the question of like, what is the interface? How do you access the data? If you ask 10 different persons, they will want 10 different versions, fully GUI, API. I just want to download the whole thing in bulk and do my own script. So, that's a problem that everyone has. That's not a virus-specific problem. The first part, I feel, is a bit more of a virus-specific problem, or at least a metagenome-specific problem. How do we deal with all this metagenome data and these metagenome assembled genomes is a big challenge. I think we have I think most people agree on what would be like the end game and what would be a good solution eventually. So we have to figure out how to get there.
I raised my hand, Joanne. Is it okay if I jump in? I just want to, Virusor was mentioned in the chat, and I guess I'll call myself the dinosaur right now in this field a little bit. This is a dream to be where we're at right now, to have this discussion of, oh my gosh, there's hundreds of thousands, millions of viruses out there that have now been sort of identified or seen across all the environments. And so I think one of the powerful things of this field is that we've been, tiny but mighty, meaning that, very few people actually get funding to do virus-focused work, particularly in environments outside of the human. And yet we've really is productivity. And I think the strength of that has been the community interactions, right? And the fact that it is an open community, we really do discuss things quite a bit, and we try to find win-win situations anytime there's overlap. And so, I think a screaming signal from the last few days is there's a few bottlenecks right now, and we have an opportunity as a field to consolidate some resources, which are minimal. Even I'm delighted, Manja, that it's a big grant, but it's still a small grant compared to how much money goes into, for example, microbiome resources. And I'm not faulting that. It's just, I think, as a group, we want to think strategically. So I would echo Simon's thoughts that, wouldn't it be great if the next phase is to get together some people who want to really try to push in this space and do it together, find a way to do it together. I have no interest in trying, iVirus, we're trying to hack things together so that people can do some analyses, but we've recognized quite a while ago how challenging these database problems are, and we're not the right group to do it. And so I'm really delighted to see some different folks who actually know something about databases here. So I'm happy to organize such a thing. I'm happy to get those kind of discussions going, and I hope we can launch from here to do that. So thanks, Simon, for kind of teeing that one up.
Excellent. Thank you, Matt.
And sorry, can I just add?
Please, yeah.
So at the moment, we don't own, so EBI, what we process and what JGI processes, just partly because of our user communities and our remits, we actually, so Nikos and I have had this discussion and sort of done the very rough estimation, and actually the intersect between two different resources, what the overlap is actually tiny, relatively. So I think we're already sort of almost doing that federated, distributed model of what we're processing. It's how we share the results, and to be honest, it sort of always comes back to the same thing. I think our fundamental limitation is just how do we get funding? But we probably need to come up with a proof of concept to get that funding in the first place. But I don't actually think we're a long way away from the mags, and I don't think we'd be that far off one another in terms of the viral sequences. And so I would be happy to have those conversations. And that's, sorry, and obviously that's not, I'm just saying between JGI, I know what the intersects, there are other resources out there. So, you know, this is not a two-way thing, it's a multi-party.
Great. Yeah, I would love to be a fly on the wall for those discussions. I think those are really important. And clearly, you know, I think another use case, well, not use case, you know, whole field of virology that's come up in the chat now is that, you know, a lot of what we're describing, these databases so far, they are focused on phages. Not all of them, certainly, but a lot of them. And we don't, you know, the thresholds and the community consensus information that's been sent out recently for viral ecology, a lot of that has been focused on phages. And so, you know, the RNA virus people are like, okay, well, we can't use a 10 kb cutoff because my goodness, like all of the RNA genomes almost are shorter than that. So that's sort of another, you know, I guess a use case that would be worth discussing when we come together to talk about databases is what are all of these sort of caveats, and are there guidelines that we can help provide to the community for how to think about best working with different kinds of data? And I think Rob was the one who said this, is that there's sort of two sides to this, right? Where there's the side of, you know, some users want a little bit of, you know, prescriptive information about, these are some general guidelines of what would be the best way to handle your particular data set. And others are like, you know, I have this specific data set, I really want to, you know, analyze it really well. And so I think meeting both of those kinds of needs with the same sets of databases and tools is a challenge. Let's see, in the chat, are there? Great. So yeah, one more thing I think from questions from the chat. This was about the actual, oh, sorry, Manja, did you want to say something first? Please go ahead.
No, I was just preparing for answering the question.
Okay.
Maybe you can ask the question first.
Fabulous. So this was actually about kind of the back-end structure, the back-end computational structure of some of these databases. And so I guess the question is are they mostly through SQL? Are they mostly through graph and vector database? Like, what does actually the back end of some of these databases look like?
I can tell for us it's a bit of both, but it's mostly a whole lot of small SQLite databases. That's kind of the ugly truth behind the scenes.
And that's how you usually start to build up something. But when it becomes larger, then yeah, you need to think if it needs to be restructured, or how far can you go with that? Yeah. But usually you start like that. That's normal, I guess.
Yeah, we have MongoDB, object stores, SQL databases. But it's not a one size fits all. We put things into different data stores based on the data type and how we think we need to access it and how we think the user community needs to access it.
Great. So kind of a fun one here. Is anybody considering AlphaFold and using AlphaFold in any of these databases?
Yes. We already are.
That's
but it's
super
expensive.
exciting.
It's expensive. That's the problem. So, I mean, we're doing it more in the mag world at the moment, but this is something that we have every intention to do in the viral world. And the other area that we're sort of doing is using the magnified protein sets for building models for interpreting cryo-EM density for colleagues in EMBL. So we've got the depth of sequence that you need for some of these things, and so we can then build the better models than you can searching other databases. So it's very exciting times.
Maybe I can add to that. I'm a passionate Go player, and you know that AlphaFold was actually an extension of AlphaGo back then. And actually, we had the same problem in the Go community. Is it possible to really go over all the games with AlphaGo back then? And actually, the community went into a way which I see may also work for virologists, that actually every person runs it on his own computer for only a couple of games and then really bring it together at the end. And I can imagine that this is also possible in such a way. But if you want a resource, a database research, which actually runs AlphaFold just on all the sequences coming in, I think that is impossible. So, but I think if you are interested in some, you have some results and then upload it, that should be possible.
Excellent. All right. We've got, if RNA viruses are under considered, what about viroids? I think that's a totally fair comment. And I think we all agree. Like, yes, I think, well, also, what about soil viruses? Come on, guys. Why haven't you made this epic, ginormous database of all of the soil viruses that we can use? I think there's a lot.
I think we will tackle that first. We will first make a soil database for Joanne, of course.
Thank you. Thank you. That was the whole
Soil's too hard. Soil's too hard. Can't assemble it.
Oh, great. Yeah, no, I think.
Yeah, I think there was also. Yeah, sorry. I mean, there's more. It's not only viruses. There's also the point about EVEs. Almost nobody really works on endogenous viral elements. This is also a huge topic. There is still this number from, what was it, 30 or 40 years ago, that we think the human genome consists of 8% of virus, retrovirus origin. And actually, yeah, maybe you can do it as a homework and try to figure out if this number is actually true. It's 30 years old. Maybe you figure out this number could be different from nowadays. Yeah, but it's not only EVE, it's also prophages. Yeah, I think there's quite a lot of things in that direction. But I think as a first attempt, it would be good to store or be able to store viruses in general, like fungal viruses are not stored somewhere. Plants, unicellular organisms having also viruses, surprise. Yeah, they are really having a problem to reach any database at the moment. So yeah, there's a lot. And yeah, viroids, of course, as well.
Well, that kind of brings up a question of who needs to be in the room for this big discussion that we're going to have. Obviously, the people working on databases, and we've established that there are limited resources, and they're certainly not going to be able to go down sort of every single rabbit hole, but we're going to have phages really well represented. What other people do we maybe want or need to have in the room for these discussions?
Plant viruses are so weird. We need the expert here. And I know you know that very well too. But yeah, phages for sure. The extreme viruses, extreme in terms of extreme environments, and that can be phages, archaeal viruses, or even eukaryotic viruses, but these are usually very atypical in terms of genome structure, so probably need them here, only the expert here, I should say. RNA viruses as a whole is its own world. Can be connected, gladly. I love RNA viruses, but yeah, it has such a different types of data and genome too that,
Well, and I guess this is kind of a related question too, is to what extent do we want, the first discussion to be, do we want the first discussion to actually be kind of focused on phages and then, expand from there? I think that's, I guess it depends on who's organizing, right? But yeah, I think we have enough to tackle with phages that, I don't know. Yeah, Rob?
Yeah, sorry, Joanne, I was going to say, I mean, it depends what the objective of the meeting is. Is it, I mean, it could be a very technical discussion about how data resources essentially do a federated data exchange, which would be very boring. I would enjoy it, but it would be very boring for many, because that sort of technical problem floats my boat as much as the sort of biological problem. Or we're trying to address the biological question. So what, you know, that will dictate who you need in the room.
I just clicked away a question from the audience. I'm sorry, so I will answer it live now. Somebody was asking if how many unknown viruses actually out there for human. So this was the question. So how many viruses are unknown for human? And I've read quite a while ago that there's an estimation of 320,000 viruses on mammals being unknown. So I don't know how much it is on viruses, but it's 320,000 different species on mammals. That's what people guess are unknown. I mean, it's an estimation, but yeah, maybe that helps. Sorry, Rob, for putting that in, but I just clicked away a question.
All right. I think we've got most of the questions have been covered. So I'd like to get maybe some parting thoughts from the panelists. What, if anything, have you either said that you want to reiterate, or do you think still needs to be said? We'll start with Simon, no pressure.
Yeah, it's a tough one. I think Matt's perspective is right. And we have, to an extent, complained a fair amount on like, well, it's complicated to deal with this data, and oh, this is all contamination, and oh, look, this is not a great genome, and oh, like sure, but also, yeah, just like thinking about where we were 10 years ago, like this is an absolutely incredible time, and the resources we can build now, and the number of viruses or virus genomes or even structure and everything we can try to get is like mind blowing. I still can't wrap my mind around it. So, you know, we have to keep this excitement. And I think, you know, every time you look at the biology behind it, or you start to, you know, manage to not get overwhelmed by the size of the data, take this small piece, or like take this, you know, specific way of looking at it, and, you know, start to look into the biology of things, it's absolutely amazing. So yes, it's a daunting task, but we are not the only one being faced with it. We can iterate and check what other communities have done. Like Matt was saying, we are a small enough community that it has always worked fairly well in terms of just like organic interactions between different groups, and different groups leading different part of the field, so that it you know, kind of by itself self-organized pretty well, I would say. And so I'm reasonably optimistic about what it will look like in the next few years, because I think we have both the incredible resource to work with, and we have enough talented people in the field that we will figure out a way forward. Won't be perfect, won't be ideal, but it will be there. So yeah, and I'm just like, I wish I could just fast forward 5 years and see where we are, because I'm really excited about this.
Excellent. Thanks, Simon. All right, Manja.
Yeah, so I think I can see that still now people are contributing what the database should have. And I just invite you, I will leave the link a little bit longer open. So if you maybe just go to bed tonight and then you cannot sleep and you just, there is something I always wanted to have. Yeah, so just put it in. We will keep it open for another week or something. And then we will, of course, share it also with all the people running databases already, because they are probably also interested in that. And so I think the job is also on the user side right now, not only on the computational side. And then we try our best to perform. Let's see what is possible and what is not possible. Yeah. So thank you, everybody, for contributing. It's great.
All right, thanks. I think my Zoom just got very slow, so we'll go on to Rob straight away.
I agree with everything that's been said so far. I think we have an opportunity now as a community. Viruses have suddenly come into the front of everyone's mind. And so I think there may be more opportunities to get the resourcing we require. So, I think we could be at an inflection point where we can really make some big strides going forward.
Excellent. All right. Hopefully I'm not appearing totally frozen, but it seems maybe I am. So I'm going to be fast.
We hear you. We hear you fine, Joanne.
All right, fabulous. Thank you. So I just wanted, since we can't let Carolyn Malmstrom speak, I wanted to read her comment out, which says, want to really urge inclusion of plant viruses in this effort going forward. There are millions of plant viruses passing through the human gut and lodging in soil, so they're not separate from the phage environment at all. And yes, I'm in a plant pathology department. I absolutely, definitely want to include plant viruses. All right, so I'll briefly close out the session part, and then maybe no pressure, Matt, after I do that. I don't know if you wanted to say anything quickly, but wanted to thank very much our panelists today for some fabulous talks. I thought that was a really rich discussion and just a great session, and we have a lot to think about moving forward. And thanks to everyone for attending. And then Matt, if you want to say any final words.
Yeah, thanks for queuing that up too. It's nice to have a minute to think about it. I just, thank everybody who's been totally engaged here. This has been wonderful to hear from so many folks in the community. And this is really the brainchild of the organizing committee. So a number of folks, they're all listed in the pamphlets and whatnot. So thanks for all the great input to create this environment. I think it was a really nice environment, be it virtual, to be able to have some discussions and to work through stuff. One thing, just remind you all that we are again going for an in-person Viromics workshop next year. It's going to be May in Columbus, and we're going to try to do a hybrid situation so that we'll be able to have kind of key, broadly interesting components that'll be available through an online interface, so it can be more accessible to everyone. That's it. Thank you, everyone, for joining us. Thanks, Joanne, and today's panelists for a great session on databases. Take care.